Hello everyone, I am Matteo Cecchetton. I'm a PhD student from the University of Genoa. This slideshow was supposed to be presented by Stefano Schiaparelli. However, due to this particular condition, I was able to present it myself. For this presentation, I'm going to present a project we've been working on the last couple of years, and in specific about the application of high-throughput sequencing technologies to the filters of the desalination plant of the Italian research base Mario Zucchelli to disclose the intra and interannual variations of the nanoplankton communities in Terranova Bay in the Ross Sea. One of the main characteristics of a sound biodiversity monitoring program is the production of fine-grained data. For fine-grained data, we mean the production of a collection of data sets or data in general, which highly represent in space and time the area and the time range investigated. This is particularly difficult as a continuous sampling effort is often required, and traditional methodologies have failed to provide an optimal solution to these particular issues. Traditional methodologies are often characterized by a variety of different kinds of technologies or methodologies. They've been demonstrated to have a low reproducibility, and they require a lengthy period of processing time with a lot of personnel involved. This is especially true for remote areas such as Antarctica, where the logistic constraints are always hampered by the environmental condition, by the extreme environmental conditions, and these logistic constraints may also be driven by financial shortcomings. Sewell and Jury in 2009 introduced an alternative method for the continuous sampling of zooplankton in the area facing the New Zealand Scott Bays, and they proposed the use of desalination plant filters. Desalination plants have been used and are present in many research bays in Antarctica for the lack, obviously, of drinkable and potable water. To produce potable water, they draw seawater constantly throughout the entire facility, and basically sampling the seawater almost every day and almost every hour of every day. This will bring a continuous sampling methodology, and at the same time reduce the costs and the period of time, and at the same time reduce the number of minimum needed personnel, as theoretically it only needs the technician of the desalination plant and the researcher. For our project, we wanted to apply high-throughput sequencing technology to this particular method, as these methodologies have been demonstrated to have high reproducibility and sensibility. And at the same time, this would furtherly reduce the costs and the processing time of the entire operation, the entire monitoring program. This program is part of the bigger TNB Code program project funded by the Italian National Antarctic Program. And for this project, we wanted to look at a correspondence between the levels of plankton biomass and the filter replacement rate. The filter replacement rate is basically it represents the lifespan of each singular individual filter, and as I'll show you later, this is important to draw out every possible other explanation of a variety of this filter replacement rate. Secondly, we explore the composition and possibly the possible short-term dynamics of the nanoplankton, nanokaryotic, and particle-attached bacterioplankton communities collected by the 5 micrometers mesh size cartridge filters of the desalination plant in 2012 and 2013. And finally, to address each potential issues on the entire sampling protocol and the following protocols to achieve, possibly, at a general scale, a standardized protocol, and ideally to propose it to the at a continental scale in Antarctica. As I mentioned earlier, the study area is located near Mario Zucchelli Station, and which is located centrally in the Terranova Bay area at the end of a small promontory in a location that is called Punta Sacchino. In the map on your left, you can see the lower red square depicting the position of Mario Zucchelli station, and the red arrows indicating the exact location in Punta Stocchino of the desalination plant intake pipe. The desalination plant is composed of two main facilities: the pump shed, which is closely located to the seawater intake pipe. Seawater intake pipe draws water from 4 meters of depth to avoid any damage from the sea ice. And from there, the seawater is drawn through a series of pipes to the main powerhouse, which is located approximately 120 meters far from the pump shed. The seawater is drawn at a medium rate of 3.5 to 4 cubic meters per hour, and the water mass in the entire desalination plant is replaced 15 times per hour approximately, or once every 4 minutes. The seawater is then drawn to the main powerhouse, and it is treated by some pre-filtration steps. The first one being a filter filled with anthracite pebbles, followed by 25 micrometer filter, and finally by 5 micrometer filter, which are the one used in the present study. These pre-filtration steps are used to remove most of the biomass, of the plankton biomass, and of the debris from the seawater to facilitate the final desalination step, which is operated by a pair of ceramic filters. In the powerhouse, the electronic logbook records the date and time of each filter replacement, and at the same time of the activation and turn off of the facility for technical purposes. This particular data would allow us to measure exactly, or have an estimate, of the hours which the filters have been filtering seawater, and thus have an estimate of the cubic meters filtered by each individual filters, which range for this particular study from 12.7 to 62.8 cubic meters per filter. 11 filters, 5 micrometers cartridge filters were sampled in between the end of January and the beginning of February of 2012, and throughout the second half of January of 2013. Each filter is approximately half a meter long and 6 centimeters in diameter, and from each filter 3 replicates were extracted using a cork borer to perform a circular cut on the surface of the filter. The first layer was removed to avoid any post-sampling contamination, and the immediately below layers were sampled for a total of 0.3 grams per replicate, which is the maximum allowed by the DNA kit used. In the picture on the left, you can see the filter sample on the 4th of February of 2012, which has been filtering for 22 hours. The extraction protocol used was the Dianese Power Soil kit from Kiogen, and the manufacturer's protocol was followed for every step except for initial incubation at 70 degrees Celsius for 10 minutes with the C1 solution in a thermostatically controlled water bath, and finally eluting the filter on the solution in 50 macroliters instead of 100 with the C6 solution. The 16S and 18S genes were amplified for 450 base pair and 550 base pair respectively of amplicons, which were then sequenced using the Illumina MySec V3 reagents on an Illumina MySec. The bioinformatic pipeline then used for the bioinformatic pipeline, we used then a variety of programs, and for the beta analysis, final beta diversity analysis, we used mainly the Philosec package in R and Chime2. Different environmental data were also taken, especially air temperature, wind intensity, and wind direction, which were downloaded from the Meteoclimatological Observatory site at the Italian research base Marzucchelli. And this data was downloaded for the automatic weather station Eneide, which is located 820 meters far from the Marzucchelli station, and was then processed using a variety of our packages. Satellite data on chlorophyll and particulate organic carbon concentration was downloaded from the NASA's Ocean Color website. This data was downloaded with, for the area shown in the lower right map at a 4-kilometer resolution, and for daily and monthly climatological data. This data was then processed with GDAL for the conversion from net CDF to the comma separated value, and then processed in ggplot2 with the ggplot2 package in R. So the first aim of this project was to look for a correspondence between the replacement rate of the filters and the chlorophyll and the plankton biomass present in the seawater. Historical data on filtering activity, meaning the duration of each singular filters for the cartridge and back filters, was downloaded from the electronic logbook from 2002 to 2019, as well as the daily water requirement. The daily water requirement is basically the consumed water rate of the research station, which is drawn from for the research and logistic activities. And as you can see, the lifespan, the filter's activity, duration of the cartridge and back filters shows a consistent decrease from the beginning of summer to the end of the summer. However, this particular trend was not observed for the consumed water rate, meaning that the research and logistic activities does not influence the rate of the filter replacement. On the other hand, the the concentration of particulate organic carbon and of chlorophyll showed a consistent decrease, obviously from October to February, meaning that the plankton biomass, meaning that as the phytoplanktonic blooms takes place, the amount of planktonic biomass increases, thus reducing the lifespan of each singular filter, because each filter clogs more simply more earlier than usual. The ordination method shows the clear distinction between the 2 years, represented by the triangles for the 2013 and by the circles for the 2012. At the same time, it showed a particular condition for both years. For 2012, for example, the first day of sampling, meaning that the first filter sampled for the time range investigated, was completely separated by the others, as shown by the red and blue, the red square and blue rectangles. And for the 2013, at the same time, the first filters correspond to the first days of the time range investigated were separated from the following ones. This has an explanation. In fact, you can see here a detailed overview of the dynamics of the filter replacement rate, the particulate organic carbon and chlorophyll concentration, as well as the air temperature and surface air temperature and the wind intensity for the 2012 and 2013 years. The green band represents the time range investigated. And if we look more closely for the 2012 dataset, we can see that it was characterized by two katabatic wind events separated by a period of relatively calm weather. And this, as you can see in the graph, the alternating blue and green bands represents the lifespan of each singular filter sampled for this particular study. And if you look again at the NMDS, the first filter was separated from the following ones, meaning that we looked at a completely different situation. In fact, the first filter had a completely different composition in terms of taxonomical composition in respect to the following filters, which were separated by the first one by the period of calm weather and were characterized by a second different katabatic event. This short dynamic was revealed by this particular technique, and the composition of the 18S dataset shows a nanokaryotic community dominated by dinoflagellates from different groups, whereas the particle-attached bacterioplankton showed the typical composition of this particular kind of bacterioplankton being dominated by alpha protobacteria and bacteria of gamma protobacteria and of bacteroidea. The 2013, on the other hand, showed no persistent katabatic event except for a couple of katabatic events. However, as you can see by the NMADS, the first days were different, were discriminated from the following ones, and you can see here that they were basically separated by the others by a period of sampling activity, which could be not sampled in those particular days. And both this year, as for the 2012, was characterized by community dominated by dinoflagellate, and this period of calm weather may show, as you can see, a turnover of the community from one particular Damphice order group, which was completely cancelled by the arrival of and the growth of another group and possibly a bloom of another group of dinoflagellates. On the other hand, the 16S dataset didn't show any particular trend, except apart from the fact that it was also recorded the same condition as for the 18S dataset for the 16S dataset, so the first days were separated from the following ones. However, the same community was observed with no particular or evident trend. This method would allow a reduced cost in the analysis, in the sampling activity, a reduced period of processing time, and for this reason it can be particularly useful for biodiversity monitoring programs, especially in remote areas with harsh conditions, where sampling is not always guaranteed and possible. The high sensibility and reproducibility of these methodologies will allow for the recovery of taxa which have never been documented in the area. For example, the Suctoria, Criomonodida, and Pseudophoria groups of the nanokaryotic community were recorded, and they were never been recorded for this particular area in Antarctica. At the same time, this particular method, by continuously sampling throughout the range period, allowed for the revealing of particular shifts, which may have we have a short period of in a short period of time at an unprecedented level, and this method can be possibly implemented and bettered and adopted at a more general scale, at a continental scale around Antarctica, as many different facilities and research stations in Antarctica rely on desalination plants for the production of potable water. And thank you. This is it. Thank you very much for listening. And if you have any question, you can ask me at my mail, matteocecchetto@gmail.com, or contact my supervisor, Stefano Schiaparelli, at this particular email. Thank you very much again, and see you next time.